Welcome to the PubMed Daily Review for April 20th, 2026. Today we scanned 1363 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Drug Effects and Patient Outcomes. These papers investigate how specific medical interventions and biological factors influence patient health and disease progression. This cluster covers 352 papers. Representative work includes the effect of empagliflozin on renal outcomes in patients with established cardiovascular disease, systematic review and meta-analysis of randomized placebo-controlled trials, multifactorial inspiratory muscle training in patients with diabetic polyneuropathy, a qualitative study, and romosozumab versus teriparatide for the treatment of postmenopausal osteoporosis, an overview of systematic reviews with direct and indirect meta-analyses. Trend 2 of 5. Cancer Disparities in Biology. This trend explores how biological factors and demographic differences influence cancer development, treatment, and outcomes. This cluster covers 188 papers. Representative work includes a cohort study on cardiovascular disease mortality in breast cancer patients with different subtypes, differences in cancer patient experience by race, ethnicity, and preferred language, a systematic review, and racial disparity in microvascular function among non-Hispanic white and non-Hispanic black men with newly diagnosed prostate cancer. Trend 3 of 5. Ecosystems and Environmental Research. These papers investigate environmental processes, impacts, and biodiversity across diverse ecosystems and human contexts. This cluster covers 278 papers. Representative work includes advances in modeling radiative transfer, heat storage, and turbulent transport to evaluate CO2, heat and water fluxes over broadleaf forests. The CanVeg2 model, evaluating the environmental impact of clinical research, a full life cycle analysis of a French academic randomized clinical trial, and navigating practical, political, and ethical challenges of long-term human biology field research, the Sure Health and Life History Project. Trend 4 of 5, Biomaterials and Infection Control. These papers explore how materials and biological processes interact to prevent or manage infections. This cluster covers 378 papers. Representative work includes ZBP1-mediated restriction of Toxoplasma gondii in both the early acute and chronic brain stages of infection, efficient sulfonation of syneotactic polystyrene membranes assessed by small-angle neutron scattering, and cell-in-shell metacells in single-cell nanoencapsulation. Trend 5 of 5, Neurological Conditions and Safety. This trend explores neurological conditions, their safety implications, and interventions for improved patient outcomes. This cluster covers 167 papers. Representative work includes diagnostic criteria and management of MELAS and stroke-like episodes, consensus-based statements, factors influencing engagement and work-related activities among people with dementia or mild cognitive impairment, a cross-sectional study, and a polypoprotein e-polymorphism and reproductive patterns among post-reproductive women. Up next, our first paper out of 5, Indigenous Gut Microbes Modulate Neural Cell State and Neurodegenerative Disease Susceptibility by Lisa Blackmer-Reynolds and Timothy R. Sampson from Department of Cell Biology, Emory University School of Medicine, Atlanta, Georgia, 30322, USA. Let's break down its key contributions. We know our gut microbes play a role in many aspects of our health, including our brain. But how exactly do these tiny organisms talk to our brain cells? This study dives deep into understanding how the native gut microbiome influences the complex world of neural cells and whether it can impact our risk for neurodegenerative diseases. To tackle this, researchers used advanced single-nucleus RNA sequencing on mouse brains. They compared germ-free mice to those colonized with specific gut microbes, like E. coli. This allowed them to map out how different microbes alter the gene expression patterns within various brain cell types, particularly focusing on immune cells. The study revealed that the microbiome has a broad impact, influencing key immune and neurodegenerative pathways in the brain. Notably, colonizing mice with E. coli triggered a specific activation state in brain myeloid cells. Even more striking, E. coli exposure worsened cognitive decline and amyloid pathology in mice modeling Alzheimer's disease, demonstrating a direct link between gut bacteria and disease severity. This research powerfully demonstrates that our gut microbes aren't just bystanders. They actively shape our brain's health and can influence the progression of diseases like Alzheimer's. This opens exciting avenues for developing new treatments by targeting specific gut bacteria. Understanding these intricate connections is crucial for future strategies aimed at maintaining brain health. Up next, our second paper out of 5, NR2F6-modified CAR T cells drive extrinsic immunogenic cell death and antigen-agnostic immunity in solid tumors by Victoria Klepsch and Gottfried Bayer from Institute for Cell Genetics, Medical University of Innsbruck, Austria. Let's break down its key contributions. CAR T-cell therapy has revolutionized cancer treatment for certain blood cancers, but its success in solid tumors remains limited. Solid tumors present unique challenges, including diverse antigens and a hostile immune microenvironment that suppresses T-cell activity. We investigated a new strategy to overcome these barriers and improve CAR T-cell effectiveness. To address this, we engineered CAR T-cells by deleting the intracellular checkpoint in NR2F6. We then tested these modified cells in challenging solid tumor models. Our approach focused on understanding how this genetic modification impacts tumor cell death and the subsequent immune response, aiming to uncover the underlying mechanisms of action. Remarkably, deleting NR2F6 led to robust and durable tumor regressions, even after the engineered CAR T cells were no longer detectable. Treated mice were protected from tumor rechallenge, including with tumors lacking the original target antigen. This demonstrated a shift from antigen-specific killing to a broader, antigen-agnostic immune memory. These findings highlight a critical design principle for next-generation CAR T therapies in solid tumors, moving beyond simple cytotoxicity to orchestrating host immunity. Targeting intracellular checkpoints like NR2F6 offers a way to reprogram CAR T cells into potent vaccinators. This kill and vaccinate paradigm holds significant promise for overcoming current limitations and achieving durable responses in solid tumors. Up next, our third paper out of 5, AAV immunogene therapy platform delivering vectorized cytokines defines a new modality for high-grade glioma treatment by Kela Kuwahara and Nicole K. Pulp from Siren Biotechnology Incorporated, San Francisco, California, 94107, USA. Let's break down its key contributions. High-grade gliomas, like glioblastoma, are some of the most aggressive and treatment-resistant cancers. Despite surgery, radiation, and chemotherapy, outcomes remain poor. Interferons show promise due to their anti-tumor and immune-stimulating effects, but systemic delivery is problematic. This study explores a novel approach, using adeno-associated virus, or AAV, to deliver interferons directly to the tumor, aiming for sustained local expression and reduced toxicity. To tackle this challenge, the researchers engineered adeno-associated virus, or AAV, vectors to express different types of interferons. They tested these vectors in sophisticated models, including patient-derived glioblastoma organoids and orthotopic xenografts in mice. A key technique was convection-enhanced delivery, or CD, for precise, localized administration of the AAV vectors directly into the tumor. The goal was to see if this localized delivery could trigger potent anti-tumor responses and improve survival. The results were highly encouraging. In orthotopic xenograft models, treatment with AAV vectorized IF led to complete tumor clearance and a significant prolongation of survival. This was achieved through localized interferon expression, which induced tumor cell apoptosis and remodeled the tumor microenvironment towards an immune-reactive state. Importantly, the vectorized interferon showed tumor-selective cytotoxicity in organoid models, sparing healthy brain cells, and demonstrated durable expression in vitro, overcoming the limitations of systemic interferon therapy. This study establishes AAV vectorized cytokines as a promising new modality for treating high-grade gliomas. By enabling sustained, localized interferon delivery, this platform effectively addresses the challenges of systemic therapy, such as poor pharmacokinetics and dose-limiting toxicities. The findings pave the way for the clinical translation of this innovative immunogene therapy, offering new hope for patients with these devastating brain tumors and potentially for other solid tumors as well. Up next, our fourth paper out of 5, a responder-informed gut microbial consortium enhances anti-PD-1 efficacy in a mouse cancer model by UK Jin Zhang and Sang Sun Yoon from Department of Microbiology and Immunology, Yonsei University College of Medicine, Seoul 03722, Republic of Korea. Let's break down its key contributions. Immune checkpoint inhibitors, like anti-PD-1 therapy, have revolutionized cancer treatment, offering hope to many. However, a significant number of patients don't respond or develop resistance. We know the gut microbiome plays a role, but we need to find strategies to harness its power. This research explores if a specific combination of gut bacteria can boost the effectiveness of anti-PD-1 therapy. Researchers first analyzed gut microbiome data from cancer patients to identify bacteria linked to successful anti-PD-1 treatment. They then created a specific 4-strain bacterial consortium called UJ04 from these identified microbes. This consortium was tested in a mouse model of melanoma, both alone and in combination with anti-PD-1 therapy, to see if it could improve tumor control and immune responses. The results were promising. While the UJ04 consortium by itself had little effect on tumors, when combined with anti-PD-1 therapy, it significantly enhanced tumor growth inhibition compared to anti-PD-1 alone. This was accompanied by a notable increase in crucial immune cells, like CD8 plus T cells and natural killer cells, within the tumors, suggesting a boosted anti-tumor immune response. This study demonstrates that a carefully selected group of gut bacteria can functionally improve cancer immunotherapy. It provides a strong preclinical basis for using defined microbial consortia as an adjunct to anti-PD-1 therapy, potentially helping more patients achieve better outcomes. The next critical step will be to see if this promising approach translates effectively to human patients in clinical trials. Up next, our 5th paper out of 5, engineering a compact high-fidelity Staphylococcus aureus Cas9 variant with broader targeting range and mechanistic insights into its activation by Satoshi N. Omura and Osamu Nuriki for Biological Sciences, Graduate School of Science, the University of Tokyo, Tokyo, Japan. Let's break down its key contributions. CRISPR. Cas9 has revolutionized gene editing, but it's not perfect. One major hurdle is the size of the enzyme, especially for delivery into cells. While Staphylococcus aureus Cas9, or SACAS9, is smaller and fits better into delivery systems like AVs, it has a catch. It needs a specific longer DNA sequence called a PAM to bind and cut. This limits where in the genome we can make edits. We need a Cas9 that's both compact and can recognize a wider variety of PAM sequences without sacrificing accuracy. To overcome SACAS9's PAM limitations, researchers used a rational engineering approach. They analyzed the structure of SACAS9 to understand how it recognizes its target DNA and identified key amino acids involved in PAM binding. By making specific mutations, they created a new variant dubbed ESA-Cas9, NNG. They then tested this variant extensively using in vitro DNA cleavage assays, followed by experiments in human cells and mice to confirm its editing capabilities and assess its specificity. Structural analysis using cryo-electron microscopy provided crucial insights into the molecular mechanisms behind the engineered variant's improved PAM recognition and fidelity. The results were impressive. The engineered SACAS9 NNG variant successfully recognized and edited DNA targets with a relaxed NNG PAM sequence, significantly broadening its targeting range compared to the original SACAS9. Crucially, it maintained high fidelity, showing reduced off-target activity compared to other Cas9 variants that also recognize relaxed PAMs. This variant demonstrated efficient indel formation and was successfully adapted for both C to T and A to G base editing. Furthermore, when delivered via AAV in mice, SACAS9 NNG effectively edited target genes in the liver, showcasing its potential for in vivo gene therapy. So what's the big picture? This study successfully engineered SACAS9 NNG, a compact and highly precise gene editing tool. It effectively expands the targeting capabilities of SACAS9 by recognizing NNG PAMs, addressing a key limitation. Importantly, it achieves this broader targeting without sacrificing accuracy, overcoming a common challenge in developing PAM-relaxed nucleases. Given its compact size and improved PAM recognition, SACAS9 NNG holds significant promise as a versatile tool for future in vivo gene therapy strategies. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.